And we are now recording. So this is the third part of the respiratory system lecture. We covered Boyle's law, as well as Dalton's law, as well as Henry's law last Wednesday. Today, we're going to explore the concept of gas exchange further as it pertains to the lungs. We're going to explore gas exchange with respect to partial pressure of the two respiratory gases, that being oxygen and that being carbon dioxide. In the next few slides, you will see certain statements about gas exchange and partial pressure. I would not spend too much time on the specific numbers. I would spend more time understanding the relative ratios, because that is what's going to drive that's what's going to drive diffusion of oxygen and carbon dioxide across that respiratory membrane. So, according to Dalton's law, the partial pressure of the respiratory gases are going to be such that respiratory gases will move from an area of high pressure to an area of low pressure. So because the whole point of the respiratory system is to bring oxygen into the capillaries that will then be part of blood that will return to the heart, this time the left side of the heart, which is oxygenated, we would expect to see high pressure of oxygen in the lungs, the alveoli specifically, and then lower oxygen partial pressure in the capillaries. The opposite would be true for carbon dioxide. So high pressure of carbon dioxide in the blood capillaries, and then lower partial pressure of carbon dioxide in the lungs. That is what will drive or set up that concentration gradient such that oxygen will move from the alveoli of the lungs and into the capillaries in exchange for carbon dioxide, which deoxygenated blood will have lots of, and that's going to go to the alveoli of the lungs so that we can breathe it out in exhalation. So a couple of factors to consider and appreciate with respect to this efficient process of gas exchange. The differences are substantial enough in partial pressure to drive diffusion. The distances in which gas exchange is very small. We know this because there's only, again, a single layer of cells in the epithelium, in the alveoli, as well as that of the capillaries. Oxygen and carbon dioxide are also lipid soluble, which is wonderful because surfactant is lipid based, and because surfactant is present in substantial amounts around the alveoli sacs to decrease surface tension due to water, it is important that oxygen and carbon dioxide are able to diffuse through surfactant, because otherwise it would be in the way. Now, because the alveoli sacs are folded in multiple ways, there's going to be overall a total surface area that we would consider large, essentially 1,506 square feet, which is about the size of a tennis court. You don't have to memorize that number. We have that fact in there so you can appreciate how much surface area there is around the alveoli sacs packed into the two lungs we have. We need blood flow and airflow to be carefully coordinated, because whenever we bring oxygen into the lungs, it needs to match the flow of blood that's flowing through the capillaries and the pulmonary circuit so that we can maximize gas exchange and respiratory gas flow. Now, the direction of partial pressure across that gradient is going to be opposite in peripheral tissues. And in order to really appreciate this, you are going to have to revisit the difference between internal respiration and external respiration, which we covered last Wednesday. We are going to move on to how oxygen and carbon dioxide travels through the blood. Now, in order to do this, we need to revisit the anatomy of a red blood cell, also known as an RBC, also known as an erythrocyte. So recall that red blood cells have lots of hemoglobin. There is a structure or a reminder, a schematic diagram here to remind you of what that looks like. Essentially 4 globin chains, 2 alpha, 2 beta, and at the center of each globin chain is going to be a heme group. At the center of it, an iron ion where oxygen will bind. Okay, so let's talk about the concept of hemoglobin saturation. Saturation referring to how many of the heme groups, specifically iron ions, have oxygen bound to it. So on the left side here, we've got oxyhemoglobin, and on the right side, deoxyhemoglobin. On the left side, this is essentially the configuration and the appearance of hemoglobin in arteries of the systemic circuit and veins, the vein in the pulmonary circuit, because that is carrying oxygenated blood. Now, deoxyhemoglobin would be found in systemic veins as well as the pulmonary artery, okay? So when we talk about saturation, we look at how many of the 4 iron ions have oxygen bound to it. And I will tell you that every time oxygen binds to iron, it makes it easier for the nearby iron ion in the nearby globin chain, makes it easier for it to bind oxygen. So getting the first oxygen to bind to that first iron at that first heme group of each oxygen molecule, of each hemoglobin molecule, is going to be the hardest. It gets easier until all iron ions are bound to oxygen. Maximum is 4 oxygen molecules per hemoglobin molecule. It's a 4-to-1 ratio, if you recall. And this would be considered a fully saturated hemoglobin. It wouldn't matter if there is more oxygen available to bind to this hemoglobin because there's no more iron ions available for oxygen to bind to. It is fully saturated. In deoxyhemoglobin, we find that there is no oxygen bound to the iron ions in the heme groups because ideally that oxygen has been let go of at the peripheral tissue so that it could go to the peripheral tissue to drive cellular metabolism. What we'd have instead around deoxyhemoglobin is a whole bunch of carbon dioxide being brought back to the heart or to the lungs so that it can be expelled from the body. For deoxyhemoglobin, a certain molecule, abbreviated here in yellow, 2,3-DPG, is going to be important. I'm just bringing it to your attention now because it's on this slide. We will discuss it in further detail before the end of today. For saturation, oxyhemoglobin saturation specifically, there's going to be a specific curve representing the easiness of oxygen as it binds to the second, third, and fourth heme group, or second, third, and fourth iron ion found in that heme group. It looks like an S, and so you'll see here towards the right side of the graph, you've got a plateau. There's going to be a steep curve towards the left side of the graph, representing the easiness of that second, third, and fourth iron ion in binding oxygen. Please know this curve really well. A lot of factors concerning the ability of hemoglobin to bind oxygen can be deduced just from this graph. What we're going to eventually move this conversation to is a concept called shift to the left and shift to the right. And what we're eventually saying here is what in certain scenarios, you will find this entire curve shift to the left, shift to the left. In other scenarios, you'll find this entire curve, shift to the right. Okay, so essentially what we're saying is if you see this curve shift to the right, then we would need more oxygen, a higher pressure of oxygen, represented by more oxygen molecules in the specific environment available in the blood to be able to saturate oxygen, in order to be able to saturate hemoglobin. In a shift to the left, you don't need as much oxygen in the blood to get the same amount of saturation. What will help us understand or figure out whether or not we have a shift to the left or shift to the right has to do with something, a parameter called P50, a parameter called P50. P50 is simply the partial pressure of oxygen, which you would find on the x-axis, the partial pressure of oxygen that's needed to saturate hemoglobin at 50%. So in other words, what partial pressure or concentration of oxygen does the blood need to have in order to have two heme groups bound to oxygen in a specific hemoglobin molecule? Just by following P50, we can determine whether you have a curve that's shifted to the left or a curve that's shifted to the right. So the easiness of oxygen binding to that second, third, and fourth heme group is known as positive cooperativity. It represents the increasing affinity for heme groups. Iron at the core of heme groups is that iron ion, the increasing affinity for that iron ion to bind oxygen. And the reason why that happens is because every time an oxygen molecule binds to a heme group, it changes the conformation of that specific globin chain, making it easier for that second, third, and then fourth and final globin chain to bind to oxygen. Okay, so let's consider 4 environmental factors that can affect how hemoglobin functions. How hemoglobin functions. One is the partial pressure of oxygen, which can also be considered the concentration of oxygen. The other is the pH of blood. Recall when we talked about blood, there is a certain pH and it's a small window that blood needs to be in order for it to function effectively. Temperature, as well as the metabolic activity found within red blood cells. Some of these factors, if changed, will cause a shift to the left. Other factors will cause a shift to the right. So before we move on to details, let's review the following. In internal respiration, so we're at the peripheral tissues, okay? We've got high oxygen, high partial pressure of oxygen in the capillaries of the systemic circuit, which is great and dandy because we need all of that to go to the cells. That concentration gradient of high to low will allow for oxygen molecules to diffuse from the capillaries into the peripheral tissue, which will drive cellular metabolism. There are two very important factors to focus on this slide. We know they're important because they're in red font for you. I highly recommend that we become very comfortable with what happens when pH of the blood decreases and what happens when temperature of the blood increases. The same result will occur. And then when pH of the blood increases, or if temperature decreases, the same result will also occur. So when you've got a decrease of pH, so the pH of blood decreases significantly below 7.4, or if the temperature of the body, like during exercise, increases, you're going to have more oxygen being released from hemoglobin, which essentially is a shift to the right curve when it comes to the saturation curve. And why this occurs is when you're exercising and you've got a temperature increase, because heat is a byproduct of cellular metabolism, we would want more oxygen being released from the hemoglobin of the blood to go to the cells, which is something that the cells will need in order for you to continue the exercise. So how does this result in a shift to the right? That was an intentional pause. The goal was to give you some time to think about it, maybe draw it out, if you will. So the reason why there is a shift to the right when there is a decrease in pH in blood or an increase in temperature is because you will have less oxygen, less partial pressure, or less concentration of oxygen in the blood. Therefore, to reach the same saturation point, let's say 50% saturation, that P50 is going to be moved to the right. So you're going to need more oxygen now to get 50% saturation. Now, if you are following that logic, then the opposite has to be true if the direction of pH and temperature moved in the other direction. So let's take now the scenario in which pH increases, rises above 7.4, and temperature decreases. Less oxygen will be released, and the entire curve will shift to the left. You will not need as much partial pressure of oxygen in the blood to saturate hemoglobin at 50%. In this slide, we show you how the difference between shift to the right and shift to the left based on the effect of pH and the effect of temperature. This is for you if you are a visual learner. So we've talked about how oxygen binds to hemoglobin. Let's now discuss the Bohr effect, which focuses on the other respiratory gas, that being carbon dioxide. And carbon dioxide is going to be found in deoxygenated blood. It's going to be found in the blood where you find deoxyhemoglobin, because there's no oxygen bound to hemoglobin, but lots of carbon dioxide as a byproduct of cellular metabolism now finding its way to the lungs by way of the heart so that it can be expelled during exhalation, breathing out. What's interesting about carbon dioxide as a gas is it actually takes several different forms as it's traveling through the blood. It will easily diffuse into the red blood cell, And red blood cells have an enzyme in bold for you here, so please know it oh so well. It's called carbonic anhydrase. And just like any enzyme, it will catalyze, speed up a certain reaction. Carbonic anhydrase will take carbon dioxide and water, and there's lots of water, right? There's lots of water available because water is the universal solvent. So when carbon dioxide and water meet, and there happens to be carbonic anhydrase nearby, this carbonic anhydrase will produce carbonic acid. And just like any other acid, in solution, carbonic acid, H2CO3, can easily release a hydrogen ion. It's the very definition of an acid. It's chemistry 101. So on the right side of this equilibrium equation, you've got a hydrogen ion and its respective anion. Please understand that without carbonic anhydrase, carbon dioxide will stay carbon dioxide and water will stay water. It's the presence of carbonic anhydrase that drives the production of carbonic acid. So we just talked about the importance of pH of the blood and how it affects the ability of hemoglobin to bind oxygen. Carbon dioxide plays an important role in buffering the pH of blood. By changing carbon dioxide count in the cardiovascular system or the availability of carbon dioxide in the respiratory system, we can increase the pH of blood or decrease the pH of blood. I want to add that this is really just half the story. While the lungs are really important at buffering pH of blood at 7.4, they are not the only organs responsible for this buffering effect. The other organ that's responsible is the kidneys, which we'll talk about more as part of the urinary system. Speaking of effect, this effect of producing carbonic acid from carbon dioxide in red blood cells is known as the Bohr effect. Do you remember when I mentioned 2,3-BPG with reference to deoxyhemoglobin? We're going to talk about it more now. So 2,3-BPG, that's a mouthful, stands for, 2,3-biphosphoglycerate. 2,3-biphosphoglycerate. Know both the long name as well as its abbreviation. You are responsible for it. In a lot of medical literature, it's also referred to as simply BPG. This molecule is created in red blood cells during glycolysis, the breakdown of basically sugar, glucose, with the presence of oxygen to make ATP molecules and pyruvate, which will eventually drive other cellular metabolic processes. So what does 2,3-BPG, what does BPG do? BPG will bind the beta chains of deoxyhemoglobin. The beta chains of deoxyhemoglobin. Beta chains only, not alpha. You can see how we can ask questions about this because alpha chains become a very appealing distractor. How I remember this is B for BPG binds to B for beta chains. Do what works best for you. This presence of BPG when it binds to beta chains will stabilize the T state. And T state simply means a tight binding structure, a tight binding structure. Seen on the right side here if you are a visual learner. Now, in the T state, the tight binding structure, it's called the tight binding structure because if you look at the openings in each of the globin chains that would otherwise allow for oxygen to sit and bind to the heme group, there's not much space there anymore. There was lots of space in the oxyhemoglobin, but not a lot of space, actually no space really, for oxygen to bind. So in a tight binding structure, 2,3-BPG will keep this deoxyhemoglobin in a state that will not want to bind oxygen. This is what will help with the compartmentalization of and separation of arteries from veins in the systemic circuit, and also make sure that we just don't that we don't undo what we just did as we're trying to properly deliver oxygen and carbon dioxide to the appropriate organs in the body. There are two scenarios in which we expect to see an increase in BPG production in red blood cells. They are if you are in higher altitude and you're having difficulty adjusting to this higher altitude. Another scenario are patients who are in the hypoxic state. So decreased availability of oxygen. It's derived from the term hypoxia, if you recall that from a previous lecture. Here is an excellent overview for all my visual learners of factors that will result in the curve shifting to the right. So here you will need more oxygen in the shifted curve to the right to reach the same amount of saturation. There will not be any cases on acidosis and alkalosis on celebration of learning number 3. They will appear in celebration of learning number 4 after we talk about the urinary system. So for now, let's just go over. So if you go over the bolded words on this slide because they are worth knowing. Alkalosis, root being alkaline, represents an increase in pH, so more than 7.6. And then an acidosis, it's a state of blood in which pH has decreased significantly, and that would be less than 7.2. So again, the pH of blood is buffered very closely. There's not a very wide window at which it can operate well. So at 7.4 plus minus 0.2, that's what most chemical panels on lab work will reference as normal when it comes to measuring the pH of blood. A few comments on the difference between fetal and adult hemoglobin. So fetal and adult hemoglobin will both have alpha chains, but fetal hemoglobin will not have beta chains. What they will have instead are gamma chains. So to summarize, fetal hemoglobin will have 2 alpha, 2 gamma chains, and adult hemoglobin will have 2 alpha and 2 beta chains. Because they have different chains, we can expect fetal hemoglobin to function a little bit differently than adult hemoglobin. Before I make the next statements, let's consider the following. In a developing fetus, there is a lot of metabolic activity going on. It takes a lot of energy to grow organs, to grow limbs, to develop the nervous system, and so on and so forth. Generally, it takes a lot of work, a lot of energy to grow. And so because of that, developing fetuses need access to a lot more oxygen. They cannot afford to be without oxygen. So what gamma chains allow is for fetal hemoglobin to bind more easily to oxygen than adult hemoglobin. It will also allow the developing fetus to more easily take oxygen from maternal blood, because the developing fetus will likely need it a whole lot more than mom. So when we compare the P50 to fetal hemoglobin, between fetal hemoglobin and adult hemoglobin, the P50 will be less than adult hemoglobin. It's essentially a shift to the left if you were comparing fetal hemoglobin to an adult hemoglobin saturation curve. On the slide, we highlight the 3 ways. You might be asked about this, so I'm highlighting it for you. The 3 forms, the 3 ways in which carbon dioxide may be present in the bloodstream. Carbonic acid, because of carbonic anhydrase. It can also be bound to hemoglobin within red blood cells, not nearly as much and easily as oxygen, but possible, or it could be found dissolved in plasma. Most commonly, at 70%, is that carbon dioxide will be transported as carbonic acid, and that is worth knowing. Next is the amount of carbon dioxide bound to hemoglobin. This form is known as carbaminohemoglobin, bolded for a reason. And then least common is just carbon dioxide being transported in the plasma. So most commonly carbonic acid form because of carbonic anhydrase, then carbaminohemoglobin, and then least is carbon dioxide traveling in plasma. Let's hone in on the difference between carbaminohemoglobin and deoxyhemoglobin. So the main take-home point here is that carbon dioxide does not bind at the same site that oxygen does. So there are other sites on hemoglobin for carbon dioxide to bind to. They'll be somewhere on the globin chains. And that is where carbon dioxide will bind to form carbaminohemoglobin. And this is important because we want to minimize the factors, things that would inhibit the delivery of oxygen throughout the blood. So we've talked a lot about the role, the interplay between the exchange of oxygen and carbon dioxide. We're going to explore that concept further, and it's known as the Haldane Effect. This concept describes how oxygen concentrations will determine hemoglobin's affinity for carbon dioxide. So as we're moving through the Haldane effect, remember that it's got to be set up in a way such that oxygen can easily diffuse from the capillaries and into the peripheral tissues, and at the same time make it easy for carbon dioxide to diffuse from peripheral tissue into the capillaries. So at the core of Haldane effect, we simply say that when you see high oxygen concentrations, it's going to increase the likelihood that carbon dioxide will unload from hemoglobin. And this comes back to eliminating factors that would otherwise compete with oxygen. And there are significant ramifications should the delivery of oxygen to peripheral tissues be compromised. In other words, the Haldane effect focuses on the importance of oxygen. We can change the amount of carbon dioxide in the respiratory and cardiovascular system by changing the concentration of oxygen in the respiratory and cardiovascular system. An example that I can give you based on something that maybe you experience at some point in your own personal lives is our scenarios when we're feeling a little tense, apprehensive, maybe that anticipatory anxiety. The tendency is to breathe less, essentially hypoventilation. And in hypoventilation, what we're subsequently doing is decreasing the amount of carbon dioxide that is leaving the lungs. And that can trigger other physical symptoms, because for as long as carbon dioxide is hanging out in the body, it will result in acidosis, a decrease in pH of the blood well below 7.2, because carbon dioxide will get converted to carbonic acid because of carbonic anhydrase. And so what we tell patients when they're feeling this way is to breathe deeply. And when we say that, what we're essentially encouraging them to do is to increase oxygen intake into the lungs, because we know that based on Haldane effect, the concentration of oxygen is what will change the concentration and unloading of carbon dioxide in the blood. And that's essentially what this graph is telling you here. Here is an overview or summary of oxygen and carbon dioxide transport. There really is nothing new I need to add on here. I would not belabor the percentages. Everything is going to be relative with respect to celebration of learning number 3. I'm going to end off today by talking about the respiratory centers of the brain. So we don't think about breathing, right? We just do it. The reason why is because the brainstem, specifically the medulla oblongata and pons, have the respiratory centers that make sure we breathe. The medulla oblongata is going to have the ventral and dorsal respiratory group, the ventral and dorsal respiratory group named based on the anatomical locations that they are in. And then the pons are going to have the pontine respiratory group. They will control the respiratory muscles based on the sensory inputs that the body is receiving. So the medulla oblongata has the VRG and the DRG. The VRG is the primary generator of respiratory rhythm. It dictates how fast breathing occurs. Because it has inspiratory and expiratory neurons, it's also what will drive forced breathing. So basically, after you breathe in, if you want to breathe even air even more in, then that would be a form of forced breathing. The dorsal respiratory group has the special role of integrating sensory information. So it integrates the sensory stimuli that's coming from the external environment, and it will communicate that with the pons. The dorsal respiratory group has inspiratory neurons only, and it's responsible for quiet breathing, essentially normal breathing, normal inhalation and exhalation, similar to what you're doing now. The pontine respiratory group, like the DRG, receives sensory information, but it has communication with higher centers of the brain. It essentially fine-tunes breathing during special scenarios like sleep, exercise, when we're trying to say something, and when we have emotional responses to certain things. Sometimes we breathe faster when we hear certain news. Sometimes we breathe slower when we are feeling a certain way, maybe sad, maybe experiencing grief, you name it. So the basic take-home point here is the VRG is the primary, it basically sets the tone. Along with the DRG, any fine-tuning in response to specific scenarios will be done by the PRG. The inputs to the respiratory centers are driven by two cranial nerves. Please know them. They are cranial nerve number 9, the glossopharyngeal nerve, and cranial nerve number 10, the vagus nerve. There are two types of chemoreceptors, so receptors that respond to changes in pH of the blood or partial pressure of oxygen, concentration of oxygen in the blood. Please know that the peripheral chemoreceptors for cranial nerve number 9 are in the carotid bodies, essentially where the carotid arteries are. And then the vagus nerve, the chemoreceptors for the vagus nerve is found in the aortic bodies. There are central chemoreceptors that will monitor changes in the cerebrospinal fluid, not blood, the cerebrospinal fluid, and they are located on the medulla oblongata. So I'm going to define two terms for you, and then I would like for you to think about, as you're studying all of this, how changing carbon dioxide levels can affect the way that we breathe. So when you have an increase in arterial partial pressure of carbon dioxide, basically, in other words, have an increase of carbon dioxide in the arteries, that results in a state known as hypercapnia, increased carbon dioxide levels. Because of that and the Bohr effect, an acidosis state may result, and what commonly causes this is hypoventilation. The opposite is hypocapnia, where you have a decrease in partial pressure of carbon dioxide. This is commonly caused by excessive ventilation. You're not keeping enough carbon dioxide in because you're breathing so much out. The result will be alkalosis, which is a term we just defined, an increase of pH of blood past 7.6. If you're breathing out more carbon dioxide and have less carbon dioxide in the blood, there's going to be less carbon dioxide for carbonic anhydrase to catalyze into carbonic acid. Therefore, you have a decrease in acid, which essentially is a more basic solution, and a basic solution is going to have a higher pH.